All right, so today we are going to talk about meiosis. Meiosis is the other type of cell division, so we've talked about mitosis. The first distinction that we should make is that meiosis is the type of cell division that is used typically for the formation of gametes. So we are talking about the sperm or the egg. And the type of cell that would do this is called a germinal cell. I used that word last week when I tried to explain to you what a somatic cell was. A somatic cell is everything but these cells. The other thing that I like to mention here is that germinal cells, even though their goal is to make gametes, still need to make copies of themselves. Germinal cells can also do mitosis. And we'll see in our next class when we talk about what happens when sperm and egg cells are made, that some of the cells will do mitosis first so that there are several copies of them, and then they can do meiosis afterwards. So don't think of mitosis and meiosis as exclusive. Just because a cell does meiosis doesn't mean it can't also do mitosis. The other direction isn't usually true. Cells that are somatic cells don't do meiosis. Only cells that are going to make gametes would perform meiosis. Now, there are two things that happen during meiosis that are different than what happened during mitosis. One of them is reduction division. And this is the idea. When cells go through meiosis, I will generally start with a diploid cell and end up with one that is haploid. So reduction division means we are reducing the number of chromosomes that are present in a cell. The other thing that is true about meiosis is that it allows for recombination. The offspring, which come from the daughter cells, are not genetically identical. And they're not genetically identical to each other or to the parent cell that made them. So we have a different combination of DNA. And one thing that I like to mention here, and it's because it will come back in other units, is that very frequently this principle of recombination is something that is considered advantageous. So not that it's not good to be able to make a copy of a cell, but if we talk about evolution and populations and generally what's happening out in the world, it's usually fairly advantageous for every member of a population to not be genetically identical to one another. That's typically seen as an advantage for the population as a whole. And since our next unit after this one is about population ecology, this idea will come back. So these are the two main principles that are going on or that are happening because of meiosis. Does anyone want to ask any questions about either of those two ideas? So the next thing that I'm going to show you is an overview of what happens during meiosis. If you have this slideshow printed out, of course you have this, but if you don't, there is a picture on page 564 of your textbook that summarizes what is happening during meiosis. One of the things that I like to point out is just this little note here. It doesn't say that specifically on the picture in your textbook, so maybe if you are writing things down yourself, you want to add that. Meiosis isn't a cycle. Mitosis is part of a cycle. But once a cell undergoes meiosis, the product of meiosis is not going to undergo meiosis again, and that's different. In mitosis, as soon as I got my daughter cell, it could repeat the process that the parent cell made. But meiosis very clearly has an end point. Once I make gametes, That's it. I'm not making another germinal cell from those gametes. They could undergo fertilization and have other things happen, but meiosis is not a cycle. Some of the things that you should notice about meiosis just as a whole, first of all, is that I start out the same way I start out when I'm doing mitosis. The cell goes through interphase G1, interphase S, interphase G2. It starts the same way. Then I have prophase, metaphase, anaphase, Tila Phase. The same names, the same 4 names for steps, which means what's happening in them is very similar. Then there's a point where I start over again. So I went through interphase, then I did prophase, metaphase, anaphase, Tila Phase. That whole first chunk is what we would call meiosis 1, where it goes through the steps one time. Then, once I have 2 cells, each of those cells is going to go through the 4 steps again. But there is no interphase in between. So I do interphase, I have my 4 steps of division, but then instead of going through interphase again, we move right into the steps again. Prophase, metaphase, anaphase, Tila Phase. This section here is meiosis 2. Now the big differences between meiosis 1 and meiosis 2. Meiosis 1 obviously had interphase. Meiosis 2 didn't. In meiosis 1, this is when we are going from 2n to n. So during meiosis 1, we reduce the number of chromosomes. In meiosis 2, we're not changing the number of chromosomes. We're starting with haploid cells, ending with haploid cells. So frequently, meiosis 2 gets described as being very similar to mitosis. Basically, the same thing is happening. Now, this is just the overview. We're going to go through and talk about what each of the sections looks like and maybe what its defining characteristics are. Before we do that, does anyone want to ask any questions about the overview of meiosis? So we're going to talk about the phases. We'll spend a little bit more time talking about meiosis 1, because meiosis 1 is the part that's considerably different from mitosis. There'll be a picture for each phase, and what we'll do is focus on what's different. I'm not going to repeat a lot of the same notes that we already took about what happened in each of the phases. So for example, prophase 1. Now prophase 1, just like prophase in mitosis, has the nuclear membrane starting to decompose. So you can see there on the picture that's happening. It has centromeres forming, or centrioles forming, with those fibers attached to them. That's the spindle. And you have chromatin condensing to chromosomes. None of that is different. All of that happens during prophase. So what we're going to focus on are the things that are not the same. The first thing that's not the same compared to prophase and mitosis is the idea of synapsis. Now, I don't want you to confuse this word with another word we'll see later on. The other word is synapse. A synapse, with an E at the end, is the space between 2 nerve cells. Synapsis is a totally different thing. So synapsis is when pairs of homologous chromosomes line up. So synapsis is saying that instead of your chromosomes and the copy that got made of them in interphase, instead of them just floating around wherever, homologous chromosomes line up. They meet up with each other. So this little section here is illustrating synapsis. there are two pairs. So I have the red ones and the green ones. This is synapsis. They're next to each other. They're meeting up. When the pairs of homologous chromosomes line up, we form a tetrad. Tetra means four. If you remember from like Science 10 when you're naming molecules, tetra means four. So in a tetrad, there are four chromatids. So let's be sure of our vocabulary here. A chromatid is a copy of a chromosome. If I look at this tetrad here that was formed because of synapsis, because those pairs met up with each other, then when I look here, I see, for example, that I have some sister chromatids. So those two green ones, for example, are sister chromatids. What that means is that they are identical copies of each other. Now, where did I get that copy? The S phase of interphase, where the DNA replicated. Now, that's not the only thing I have in my tetrad. I have sister chromatids, so the two green ones or the two red ones would be considered sister chromatids. But if I were to indicate these two, one of the red ones and one of the green ones, they are non-sister chromatids. So what does that mean? They are the same chromosome. So for example, if this was the X chromosome, these would all be X chromosomes. But we would have different alleles. That means different versions of the genes that are on it. So if I was to indicate one red one and one green one, that is a non-sister chromatid pair. Now, since they are right next to each other, there's a phenomenon called crossing over that can happen. So when you take a look at the second picture, I'm going to highlight this portion right here. One of the legs or arms, depending on how you want to describe it, of the red chromatid and the same portion of the green one crossed over each other, just like you would cross your legs or cross your arms. When that happens, the result, like you can see in the third picture, is that some DNA gets exchanged. When that DNA gets exchanged, we have what is called crossing over. A portion, definitely not the entire thing, but a portion of a chromosome, a portion of its genetic information is now on the other chromatid. So crossing over increases recombination, or let's say variation, because now your chromosomes might have a slightly different information than they started with. Now, crossing over is only something that actually causes an effect when we are talking about non-sister chromatids. If they were two sister chromatids, since they have identical DNA, it wouldn't matter if they crossed over because the information would be the same on both of them. Since the red one and the green one are not identical, there's a possibility that the information is not the same on both of them, which could lead to recombination of characteristics and more variation. Now, does anyone have any questions that they would like to ask about synapsis, tetrads, non-sister chromatids, or crossing over? So this is what's different Other than that, like I said, prophase has the same characteristics. Centrioles, spindle fibers, nuclear membrane decomposing. So you would continue to look for those key things if you were trying to identify which phase of the cell cycle it was. The next one is metaphase. Metaphase is always when the chromosomes line up along the equator. I purposefully chose a picture here that had cells aligned vertically so that you didn't think it had to always be horizontal. A circle is a circle, so anywhere can be the middle. Now in metaphase 1, just like in metaphase during mitosis, chromosomes line up at the equator. But the thing that's different here is that pairs of homologous chromosomes line up in tetrads. So those two words there are really distinctive. I don't just have my chromosomes lining up one at a time along the equator. I have them lining up in pairs so that I have a bunch of tetrads. A tetrad is really, really distinctive. If you see a group of 4 in a row, you are definitely in meiosis 1. You are not in mitosis. You are not in meiosis 2. So the tetrad itself is very distinctive. Now the thing that you need to know about metaphase 1 is a phenomenon called independent assortment. And it means that chromosomes line up randomly, which means there is no regard for their origin. And I'll explain what that means. Let's say that on this picture, the chromosomes that are colored red come from mom, and the chromosomes that are colored black come from dad. When we say independent assortment, we mean that the chromosomes that you got from your mother do not all have to be on the same side of the equator. In version A, you'll see there's one red one on the left, one black one on the right, and then a black one on the left, a red one on the right. And in version B, both red ones are on the left, both black ones are on the right. Either of those is possible. So the chromosomes do not have to line up by origin, which means which parent they came from. This provides further genetic variation because it means that when you make gametes, they will not be identical to the gametes that made you. They will have a completely random assortment, some genes that were from maternal origin and some genes that were from paternal origin. So the chromosomes do not have to line up in a particular way. Other than that, they need to be in pairs of homologous chromosomes. But other than that, it doesn't matter which one goes on which side. Now those are the things that are distinctive. Metaphase generally is chromosomes lined up along the equator. You should always see centrioles, which are these guys, and you should always see the spindle fibers coming out, attaching to chromosomes that are lined up along the equator. So that part has not changed. What has changed is that they're pairs lining up in tetrads, and that there's independent assortment. Does anyone have questions about metaphase 1? So then we have Anaphase 1. Anaphase, just like in mitosis, is when chromosomes or chromatids are pulled apart by the spindle fibers. So you still have centrioles attached to spindle fibers. The spindle fibers are shortening, just like they were in mitosis, pulling chromosomes to one pole or to the other. So that part has not changed. What has changed here is this. Homologous pairs separate, not chromatids. And this means that the centromere does not divide. That is a very visible clue that you are in Anaphase 1. So when you look at what is being pulled apart, if you see two chromatids, and these are two sister chromatids, still attached, and they're being pulled to one side, that means that you have the pair separating, not the chromatids are staying together, and that you definitely still have a centromere that's attaching those two sister chromatids together. So it's really important that you recognize that we are not pulling apart chromatids, the identical copies. We're pulling apart pairs. So we could say in this example, these are the two versions of chromosome number 1, let's say that the cell has, and these are the two versions of chromosome number 2. We are separating pairs of homologous chromosomes, but not the identical copies that were made. Now, does anyone have any questions that they would like to ask here about Anaphase 1? So I'm going to pause. So this diagram is representing two possible scenarios of what is called non-disjunction. Non-disjunction is when chromosomes or chromatids do not separate when they are supposed to. So we're talking about this now because anaphase is when chromosomes or chromatids are supposed to separate. Now you see I wrote chromosomes or chromatids, and that's because this could be happening during Anaphase 1, which we just saw was when chromosomes separate, or it could happen during Anaphase 2, which, very similar to mitosis, is when sister chromatids pull apart. So the picture on the left is representing what would happen if we were talking about Anaphase 1. At the top, we're starting with a cell that has 4 chromosomes. So 2n equals 4. That means n equals 2. Now remember that a cell that's undergoing meiosis will have already gone through interphase, and there will be a copy of each of the chromosomes. So I have 1, 2, 3, 4 chromosomes plus an identical copy of each of them, but I still only have 4 chromosomes. This highlighted portion here is representing non-disjunction. Now, a little disclaimer for you. Lots of you know that I teach in French and in English, and very often my poor computer does not remember or does not even know which language I'm trying to type in. So if you ever see on a sheet or something a word that appears to be misspelled, it is never a trick. I am never trying to fool you by misspelling a word. I literally did not notice because my brain is like, yep, that's a word. I'm not thinking about the right language. So for example, in French, non-disjunction is the same word, except that the U is an O. So if you ever see that, I'm not trying to fool you, I mean non-disjunction. What happens then is that non-disjunction happens in Anaphase 1. So instead of those pairs of chromosomes being pulled apart, notice how this pair right here all gets pulled to one side. If the cell then continues to divide, so that's Anaphase 1, it'll finish doing meiosis. It'll go through prophase, metaphase, anaphase again. And the result will be, at the end, there are 4 daughter cells. These are the gametes. And they are supposed to be haploid. They are all supposed to have N chromosomes, but they don't. 2 of them have an extra chromosome. So N plus 1, in this case, since N was 2, N plus 1 is 3. 2 of them will have N minus 1. So they will have 1 less chromosome than they are supposed to. Now the problem is gametes are supposed to go on and make the next generation. So when these gametes move on to fertilization, then whatever organism is being made is going to have the wrong number of chromosomes. Now, nondisjunction can happen in Anaphase 1, which is the one we just saw. Nondisjunction could also happen down here in Anaphase 2. So in Anaphase 2, it looks very similar to mitosis. Chromatids, which are attached by a centromere, are supposed to come apart, but they don't. If it happens in Anaphase 2, we should get 2 normal cells, because only one side of the story will be affected by this nondisjunction. So we should have 1 cell that has 1 too many, 1 cell that has 1 too few, and 2 that are normal. Now, in the big picture, generally when we're going to ask about nondisjunction, especially diploma-style questions, I don't see them frequently ask, was it Anaphase 1 or Anaphase 2? What you need to know is that nondisjunction happens during anaphase. That is the key point. That is the main idea. So if a gamete has the wrong number of chromosomes, anaphase is the phase that was affected, and nondisjunction is the name of the process. Now, would anyone like to ask any questions about nondisjunction? So I'm going to remind you of something that we actually already briefly saw when you made human karyotypes. And the thing that I'm going to remind you of is, what is it called when someone has 1 too few or 1 too many chromosomes? We talked about, or we saw on your karyotypes, a bunch of people that had the wrong number of chromosomes, and we're just going to be a little bit more specific with that vocabulary now. So the consequence of nondisjunction are gametes with too many or too few chromosomes. Now you'll notice in basically any source you look at, too many means you have 1 extra. Too few means you have 1 too few. We would not talk about N plus 2 or N minus 2 or N plus 3 or N minus 3 because the probability of an organism surviving from a gamete that had that big of a difference in chromosomes is very low. Even the conditions that we do know of are incredibly rare. A monosomy is what it's called when you have too few chromosomes. So this is our N-1 scenario. So if nondisjunction happens and a gamete is produced with too few chromosomes, and then that gamete gets fertilized and an organism results from it, you would have, in a human, 45 total chromosomes. So a good example of this type of syndrome is what is called Turner's. Turner's syndrome is someone who has 22 pairs of autosomes and one X chromosome. So not two X's, not an X and a Y, just an X. This person would be genetically female, but they have a monosomy. They are missing one chromosome. So mono means one. So normally we have chromosomes in pairs. Here we have a chromosome that's all by itself. So this is where we're getting the idea of mono from. We have a chromosome that is all by itself, that is non-paired. So Turner syndrome would result generally in someone who's genetically female. It could result in infertility. It usually results in smaller size, slower development, just generally delays in a lot of the developmental features, especially the ones related to reproduction, because now you have one X chromosome instead of two. Turner syndrome is the only one where I have actually personally known somebody who had Turner syndrome. You are perfectly capable of having a life that you can live. Generally, development is a little bit slower, which usually means learning is a little bit slower. But other than that, you could live a regular length of life and be perfectly happy, even though you have this monosomy of chromosomes. Now, a trisomy, where tri means three, is, I guess, the more common of the two. There are more examples of syndromes where someone has a trisomy. A trisomy is when you have too many chromosomes. In a human, this would mean you have 47. Now, would it theoretically be possible to have a gamete that had an extra one go together with another gamete that had an extra one and make someone with 48 chromosomes? Theoretically, yes. Would that organism survive? Doubtful. Some examples of trisomies would be Down syndrome. It is trisomy 21. That means that a person with Down syndrome, they could be male or female, there's no real distinction there, they have 3 copies of chromosome 21. Other than that, they have all of the other chromosomes. So they would have 2 X's, or an X and a Y, and the other pairs of chromosomes would all be in 2s, but there would be 3 copies of chromosome 21. There are other syndromes. Edwards syndrome is one. It is trisomy 18. There's one called Klinefelter. That is someone who has 2 X's and a Y. These 3 were all examples on the karyotype sets that you had. So some of you had Klinefelters, you had 2 X's and a Y. Some of you had Edwards. So if you said your trisomy was 17 or 18, sometimes people mixed up which one was longer and which one was shorter. Or if your trisomy was near the end where you had 3 of one of the really tiny chromosomes, all of those are possible results or possible outcomes of non-disjunction. Klinefelters, maybe I'll just explain. A person who has Klinefelters is genetically male, but their secondary sex characteristics, things like facial hair and tone of voice, those types of things, tend to be more female-like in characteristics. So less body hair, higher pitched voice, that type of thing. Now, does anyone have any questions that they would like to ask about these troubles that happen? So this might be, I'm just going to point out, this might be something that if you haven't signed up for your article talk, this might be something that interests you. There are several other disorders that you don't need to know specifically. One of them is called super female, XXX. That's another condition that exists. There's one called Patau syndrome. There's another one called cri du chat, which means the cry of the cat. But there are several genetic disorders which result from non-disjunction. So that might be a topic that you could find an article about. So we're going to finish up Meiosis 1 with Tila Phase 1. Now, Tila phase looks the same as it does during mitosis. For example, you would see the cell pinching if it were an animal cell. You would generally see the nuclear membrane reforming. But very distinctively, you should see two nuclei. Chromosomes grouped on one side, chromosomes grouped on the other side. So when you were looking at allium root tip mitosis, where you could see two clear nuclei, that was your hint that it was telophase. Now, one of the things that's important here is that not all cells do this. Sometimes when a cell is undergoing meiosis, rather than splitting in half distinctively right here, it waits until it gets to the end of meiosis 2. Another thing that is distinctive here is that cells go to meiosis 2, which would mean prophase 2 after, not, absolutely not, interphase. So normally when a cell goes through telophase, it will divide in half and then go back to interphase. That is not what is going to happen here. Now at the end of telophase 1, like I mentioned before, each daughter cell is haploid. That means they each have N chromosomes. So at the end of meiosis 1, we have done reduction division. We took a cell that was diploid, we cut its chromosome number in half. Now that's the end of meiosis 1. Meiosis 1 is where there's a big difference. Meiosis 2 looks basically identical to mitosis. There's very little difference. So on the next page, I have just a few pictures representing meiosis 2, and you'll see that they look very much like the photos of mitosis. So in meiosis 2, it is very similar to mitosis. So I'm just going to point out a few things that you would look for. So here's a nice picture of prophase. Prophase of meiosis 2 and prophase of mitosis don't really look that different. Hey, you have your nuclear membrane decomposing, you have chromosomes being visible. I would say the only time you could tell that you were for sure in meiosis 2 just by a picture is if you can see evidence of crossing over. That's the only thing I have that tells me that this is meiosis 2 and not mitosis. Now down here is metaphase. You'll see again, it doesn't look different than metaphase and mitosis. You have chromosomes lined up along the equator. Again, the only thing that would distinctively tell you that this was meiosis and not mitosis is if you see some evidence that crossing over occurred. So I could ask you on a test, here's a picture, just one picture. Which stage of the cell cycle is this cell in? And there are some examples where you could tell me that it was in meiosis 2 or mitosis, and you might be right either way, because there's no way to tell the difference. Now the next set of pictures is what anaphase could look like. Now there's two pictures just reminding you that in anaphase, independent assortment still happens. So the chromosomes could be lined up either way. So on the top, it's showing you, if I was to divide my cell in half, That the red one would go on the left, the black one would go on the left, the red one with the black tip would go on the right, the black one with the red tip would go on the right. On the bottom, it's showing you the opposite scenario, just trying to remind you that there are two possibilities for what happens. At the end of meiosis, you have 4 daughter cells. So we'll call this Tila Phase / cytokinesis. Very often, telophase and cytokinesis are represented by the same picture when we're looking at meiosis. And the only thing that's different at the end of meiosis 2 is that each of these daughter cells is haploid. They have 1 of each chromosome rather than 2.